Okay, so welcome to the poster flash session, which is one of the funnest parts of a very nice tradition for this workshop. So those who don't know what it is, so this is an opportunity for the students to advertise their posters. So many people will come and hear about the work. So every one of you get 3 minutes precisely. Okay, so you have to, and when these 3 minutes will over, so we have a timer here, but I ask the audience, when you hear that the timer is off, you need to make as much noise as you can so the speaker cannot continue. Okay, okay. And for the presenters, so this is part of your evaluation, and this will you will get points for which will count towards the prize or prizes. And one last thing. So I will call the names of the speakers, but please, you should know the number of your the number of your posters. So I will say each time what's the current number, and you need to. The next in line should be prepared to switch as fast as you can. Okay? Everything is clear? Questions? Okay, let's start. So the first presenter will be Asaf Dergachev.
איפה המצגת? יש פה איזה פולדר. פוסט פלאש.
Who is number 2? So number 3, please be prepared.
Okay. Hello, thank you for the opportunity to present a part of my PhD work. I'm going to tell briefly about synthesis and evaluation of novel antifungal compounds that may have promising application in future targeted nanomedicines. Amphotericin B has been considered for many decades as a gold standard drug against a broad range of human fungal pathogens. It's highly potent but also very toxic systematically. Additionally, its administration is made difficult due to very low aqueous solubility. Despite existence of a liposomal form, which is very expensive, there is a real need for a novel potent antifungal drugs in clinics. When we started our work with amphotericin, it was clear for us that we would like to introduce some chemical modifications to make it more soluble without damaging potency, to reduce animal toxicity, and to enable a free terminal amine group on the linker for further conjugations with our targeted nanoparticles. In collaboration with Dr. Alex Spielman from Technion, we synthesized amphotericin derivatives conjugated to PEGs with different lengths. We started our synthesis with PEG that has an NHS group on one end and amine group protected by FMOC on the other end. In the following step, we performed different chemical modifications on the carboxylic group and tested how it affects the toxicity against mammalian cells without damaging the antifungal toxicity. The longer PEG we tested has 44 PEG units, and the shortest one has 4 units. Finally, we found the best combination of PEG length and modification, which we cannot publicize here until IP considerations. Here you can see biological evaluation of our compounds in vitro by examination of minimal inhibitory concentrations on Candida albicans and Aspergillus fumigatus. You can see that 2 of our derivatives have potency pretty similar to that of amphotericin, and that they are also about 1,000-folds more water-soluble. We tested the mix on additional fungi as well and showed that our compounds show efficient antifungal activity against a broad range of pathogenic fungi. Toxicity study in vitro has shown that our compounds have no hemolytic activity, while amphotericin resulted in high hemolysis. Mammalian cell viability assays show that compound C2 have at least 150-folds reduced mammalian toxicity comparing to parental drug. A toxicity study in ICR mice has shown that, in fact, we cannot reach the LD50 values for our compounds that makes the LD50 at least 20 folds lower than Amphotericin B. Finally, we performed an in vivo efficacy study. We divided our mice to 6 groups and infected them with Candida albicans. Thank you.
Okay, so next is Dmitry Rivkin. And then we should try to be in time.
This is working? Yes? Okay, start. לא, שנייה. Okay, טכני. Hello, my name is Dmitry Rivkin. I'm a master student in the David Sprinsak lab. My work is about a platform for image analysis of cells, and it's based on the Liat Zilberstein work. Now, for introduction, okay, the work is about, the laser not working, okay. The work is about looking at the cochlea at the specific displays, okay. This is the intersection, and you can see there are 2 kinds of cells, hair cells and the supporting cells. The picture from above is this level of the cells, okay. Now, in red you can see the hair cells, and the green is the boundary, and not without color is the supporting cells. Now, we can see that if we see of the development of the cells, in the embryonic day 15, the cells didn't differentiate, and you don't have a pattern here. But in day 17, you have a specific mosaic pattern that this is what we're looking for, and this is what interested us, and we try to understand the processes that influence on the pattern. Now, what I did is the platform for that you can extract and track the cells, and you can extract all kinds of data, such as the area of the cells, neighbors of the cells, and etc. You can control the tracking, the filters, and that's it. If you want to hear more, you can come to station number 3 and go.
Okay, next is Dmitry Tachinsky. And please try to speak to the microphone so they will hear you.
Hi, my name is Torchinsky Dmitry. I'm from Yuval Evenstein's lab at Tel Aviv University from the Faculty of Chemistry. I want to thank you very much for the opportunity to talk to you today. And I will present to you very briefly our work, which is quantitatively sizing femtogram amounts of double-stranded DNA by single molecule counting. So the concept of this work is we're using an intercalating dye, which is a dye that's which paints the DNA by its length. I mean that the molecule of the dye inserts itself for several bases by the proportionality by the concentration of the dye, and there is a maximum limit, but there is a linear proportionality between the intercalated fluorescence intensity and the length of the molecule. We use a reference standard, which is used as internal size calibrator, and then we image the samples in an epifluorescent microscope, and by single molecule counting, image analysis, and mathematical calculations, we find the length distribution of the sample. So the raw image of, as it's seen in our system, is like this. You see many, many DNA molecules. Each dot or line is a fluorescent DNA molecules. The standard molecules are the molecules that shine in both channels. And we do image analysis and classify the molecules by reference and the sample molecules. And after some manipulations, which I can explain to you if you come to see my poster, we received these results. And this actually represents a commercial 1 kb DNA ladder. And we sampled it. We sampled around 200 femtograms of DNA. And on figure B, you can see the comparison to the theoretical relative amounts of each population in the sample. It's in green. Our results are in red. And the top-of-the-art machine, which is the bioanalyzer in this aspect, you can see that the results for 100 picograms, 1 nanogram, and half nanograms compared to our results. And if you want to hear more how we do the internal size calibration and stuff like this, so come to see my poster. It's number 4, and I will be glad to explain to you all. Thank you very much for your attention.
Next is Ehud Heimer.
Hello everyone, I'm Ehud from the Vitro lab. I'll present to you my physics work. The notch and delta are two proteins that undergo complex dynamics on the membrane. When they get in contact, they produce cell-cell signaling that activates one cell and creates modification in gene expression of the activated cell. This causes the cell to differentiate into a certain type of cell, and it inhibits all other cells surrounding him, so they won't differentiate into the same type of cell. So we'd like to know more about the delta dynamics on the membrane, and this would entail diffusion, endocytosis, and exocytosis. Endocytosis and exocytosis are simply recycling the delta over the membrane surface. We tried to measure it using FRAP, and we got a large variability in different cells. So I developed a camera-based 2D FCS. FCS is fluorescence correlation spectroscopy. It uses the fluctuations around the thermal mean to probe, or it uses the cross correlation between fluctuations in different times in order to probe those parameters. And we also use TIRF, total internal reflection fluorescence, which uses evanescence waves. And we also use the fluorine fluorescence tag, which is a pH-sensitive tag, which allows us to get more signaling from the membrane itself and not from the inner parts of the cell. So thank you very much. If you're interested and would like to know more, I'm poster number 5. Thank you very much.
So number 6 is Eitan Vinograd.
Hello, I'm Eitan. I'm a student in Ori Chesnovsky's lab, and this work is done with collaboration with Fernando Patolsky's group. So our scientific goal is to develop and characterize silicon-germanium heterostructures which will have direct bang gap and strong optical absorption. So why is absorption spectroscopy so important? So it gives us the electronic structure of the materials. It can tell us what the band gap size and shape is, and type, and it enables us to detect small molecules. Fluorescent can be an alternative tool, but not all molecules fluoresce. So absorption spectroscopy for nanoparticles is usually done on an ensemble of particles, but the problem is that this particle will give us this type of spectra, the other particle would give us this type of spectra, and the third particle will give us this one. And the resulting spectra will be just an average, which tells us nothing about how each single molecule in the ensemble, what's its properties. So why is it so hard to check the spectra for a single nanoparticle? So for fluorescence, the challenge was only achieved in about the 1990s to detect a single fluorescent molecule. And for absorption, the challenge for detection of a single molecule was only achieved about less than 5 years ago. And that's why. So for fluorescence, we have a very low signal on a zero background. So we just need a good enough detector to be able to detect the photons from this fluorescent molecule. But for absorption, we have a very low signal on a very high background. And the light attenuation is about 10 to the minus 5 or 10 to the minus 7, depending on the molecule. And it's very hard to detect the small amount of light missing, which is much lower than our light source fluctuations. But we don't want only to detect a single molecule. We want to know its spectra. And with our home-built system, we can actually do this now. So this is a spectra from a single silicon nanowire, 80 nanometers diameter. And we intend to do even better soon. So if you want to know how we do it, you can come and listen and see our poster, number 6. Thank you for listening, and I'll be happy to.
Number 7 is Elad Eisner.
Okay, good evening, everyone. My name is Elad Eisner. I'm a PhD student in the group of Tal Ellenbogen in the Electrical Engineering Department. I'll present today our work in strong coupling in complexes of aluminum nanoantennas and thank you, and molecular J aggregates, signatures of emission from the hybrid states, and polarization from those. So in metallic nanostructure, the electron gas, the oscillation of the electron gas can give rise to specific resonances. Those are called localized surface plasmon modes, and they depend on the size, shape, and type of the material. In our experiment, one of the complexes we manufacture, of course, in the nanocenter, is aluminum nanodiscs. As you can see on the left, this is the transmission spectrum. For each diameter, we manufacture arrays, and for each array, different diameter, and those have different resonances across the entire visible regime. Even we can go down to the UV. On the right, you can see beautiful transmission images that show nice colors. So one thing in the field that wants to be done is to be able to control light actively on the nanoscale. So what happens when vacuum fields of those localized surface plasmon resonance and excitons, which are electron-hole pairs, in our case is molecular J aggregates, interact strongly. So when that happens, hybrid exciton localized surface plasmon quadiparticles are formed. Those have really interesting and new properties of mixed light and matter particles. And they can be used to achieve active control of light on the nanoscale by using the excitonic part as a handle to reach those hybrid states and manipulate them. So in our experiment, we observe a huge vacuum Rabi splittings which corresponds to energy transfer between the modes on a 10 femtosecond time scale. We saw emission from these hybrid states, and also with the nanorods complexes, we had polarized hybrid states when in one polarization we had the hybrid states, and the orthogonal one, it remained uncoupled. So if you're intrigued, come and meet me in poster number 7. Thank you.
Yeah, so now we'll have together number 8 and 12, Gur Lubin and Yaakov Ben-Dor. Well, sharing posters.
Good evening. I'm Jacob Ben-Dov, and this is Gur Lubin. We are finishing, each of us, our masters in engineering. I'm under the supervision of Professor Yael Hanin, and Gur is under the supervision of Professor Yael Hanin and Professor Ori Chesnovsky. We will present 2 different posters in semiconductor nanorod carbon nanotube biomimetic films for wire-free photostimulations of retinas.
So our motivation is artificial vision, or enabling blind people to see using retinal procedures. The concept is very similar to Geordi from Star Trek goggles or Steve Austin, and the concept is to elicit neuronal action by electrical response in response to illumination. Sorry, by electrical stimulation in response to illumination. Now, this is not science fiction anymore. To the right, you can see a couple of examples of retinal implants actually implanted in blind people's eyes and enabling them some vision restoration. This is still a new technology, so there are some limitations. Mainly, the resolution is not high enough. There's a need for curious wiring, and the devices are rigid. So our concept is to take carbon nanotubes and combine them with quantum rods. So the carbon nanotubes adhere really good to the neuronal tissue, and they also establish a good electrical conjugation. The quantum rods add to the hybrid the light sensitivity. So once we illuminate it, we achieve charge separation at the interface and activate the neurons. So come to my poster, and you can see our successful in vitro stimulation of a blind retina. And we can also discuss the underlying mechanisms by studying the fluorescence and fluorescence lifetime, as well as the photo response of different nanocomposites.
Thanks. So,
Poster number 8.
yes. In my poster, I will present photoelectrical, photocurrent, and photo voltage measurements of test films, more interesting, of PDMS flexible implants, the same kind that were implanted in vivo in a rabbit, as you can see in the lower pictures. I also present an electrical model, equivalent electrical model, and I invite you to come to my poster number 12, and we both invite you to posters 8 and 12. Thank you.
So next is number 9, Guy Herstig.
Hi, my name is Guy Herstig from Shachar Richter's lab, and I will present today a simple method to transfer large area, single and multi-layers, using adhesive material. So when synthesizing nanoparticles under certain conditions, you may get perfect layers with high density, but the downside is that you might get a surfactant layer underneath it with aggregates, and so the method that I will present shows one solution to this. So the method is very simple. And it starts by synthesizing the nanoparticles on top of the origin source. And then you make another chip, another substrate with your polymer or material that is adhesive. You can coat it, you can evaporate it. There are many ways. You can do it on ITO, on silicon oxide. Then you press the two together, and you can do it either by your hands, or if the layer is very delicate, you can do it by a nano-imprint lithography machine. Then you lift off by the solvent, and you will get a very, by the conditions of the pressing and your destination chip, you can either get monolayer, mixed layers, or multi-layers. For electric measurements, you can evaporate contacts, and of course, measuring. So this is one result of a large area transferred multi-layers of copper oxide nanoparticles, and it was done by a heated indium layer. Another result is by a spin-coated P.PSS that was half-baked. You can copy transfer a mixed layer of single layer and multi-layers. And indium without heating, you can transfer single layer, most part of it. And the method was intended to measure photocurrent of 1 layer of copper oxide nanoparticles, but it is not limited to that. So your imagination can take you wherever you want. And so if you want to hear more, you can come to my poster number 9. Thank you very much.
So now we skip a few, and next is number 13, Katrina Kulov.
Hello, my name is Catherine. I'm a PhD candidate at the Physical Chemistry Department, and I work under the supervision of Dr. Tal Schwartz. In my poster, I will show you how we use hybrid Plasmonic-Photonic structures in order to mediate long-range energy transfer. Okay, so FRET is a common mechanism that involves a donor. It stands for first resonance energy transfer, and it involves a donor molecule in an excited electronic state, transferring its excitation energy to an acceptor molecule through non-mediative dipole-dipole interaction. The disadvantage of this mechanism is that the molecule should be only a few nanometers apart. In our innovative mechanism, we mediate long-range energy transfer by coupling between surface plasmon and cavity photons. A cavity is an optical resonator that's made of 2 partially refractive walls, and it can capture photons with a certain wavelength due to constructive interference. We can control the resonant wavelength by adjusting the cavity dimensions. Surface plasmons are evanescent wave of electron oscillations. They propagate parallel to the surface, and we can control the resonance wavelength by adjusting the thickness of the metal and the refractive index of the dielectric material. If we build our cavity from metallic walls, we'll have the photonic mode inside the cavity, but we will also have the plasmonic mode outside the cavity. And we fabricate our sample such that the top mirror is thin enough so there is a leakage of the plasmonic field inside the cavity, and we adjust the cavity dimensions to reach to a strong coupling between the photonic and the plasmonic mode, so there is a resonance between them. And we use it to, we depositing donor molecules where the plasmonic mode exists, and acceptor molecules where the photonic mode exists. Okay, and we use this interaction to try, when we excite the donor molecules, they will emit their energy to the photonic mode, and then through the strong coupling between them, the energy will enter the cavity to the acceptor molecules, and eventually we'll get the emission from the acceptor molecules, and this is the mechanism. We use the fluorescein and rhodamine B molecules.
So next is number 16, near Turco.
Hello, I'm Nir from Nati. Oh, man. Okay. Hi, I'm Nir from Nati Shaket's group from the Biomedical Engineering department. On my work, I've applied gold nanoparticles to biological cells. Gold nanoparticles are particularly interesting due to the fact that they induce the photothermal effect, which is the effective transformation of light energy into heat. Let's say we have a mixed biological sample, and we want to get rid of certain cell population. The photothermal effect and the gold nanoparticles may help us in that. You see here the diluted cells, which were specifically diluted. The other cells were intact. How do we do it? Using interferometry, we capture the phase of the sample consistently, like you see here, and under weak excitation of the photothermal of the nanoparticles, the heating around them form unique phase signals, which gives us the photothermal imaging. Under stronger excitation, the heating around the nanoparticles may actually damage the cells and cause their eventual death. Our work combines the two modalities, and our vision is to use both modalities to photothermally to form photothermal therapy under controlled measures. Here you see biological cells, and the beam, the excitation beam, was located in the middle, but this is more important. Well, here you see a lateral scanning of the beam. The field of view is constant, and the beam just travels through the space. You see the cells that are ablated, they are deformed at best, they get complicated and just iffy. It happens like this, it may happen like this, and like this. Now the beam is laterally scanning the entire field of view, which resembles this. So if you happen to have a cell population you want to get rid of, or you just want to know more, come and see me at 16.
So next is number 18, Shai Kerenzu.
Okay, so good evening, everybody. My name is Chaoul. I'm a PhD student under the supervision of Dr. Tal Engelmogen in the electrical engineering school. And I would like to talk to you about the work we've been done of construction of nonlinear metamaterials where we can control them. This is a nice day to do this because today it is published in Nature Photonics, so it's a great way of doing that. So in our lab, the nanoelectro-optics, we study interactions of matter with light. So usually most interactions are linear, meaning that if you shine it with a specific wavelength or frequency in other way, the output will be at the same wavelength. So if you want to do anything else, we go to the regime of nonlinear interaction, and nonlinear materials actually enable us to do light wave mixing. Which means we can take, let's say, a certain frequency and double it, or even make 3 multiplied by 3 or even more. And even you can take two different frequencies, sum them or subtract them, and get a new kind of wavelength. Like you can see here in this picture. You can build from this many things like optical amplifiers and oscillators. You have the effect of self-focusing and many more nice effects. And this is actually very applicative since laser technology is based a lot on this kind of effects. Many things are done in optical communication and even in quantum computation. So what we're trying to do is take this kind of phenomena and take it to the nanoscale. For this, we use metamaterials. Metamaterials are actually artificial materials with tunable and engineered optical properties, where we can construct something from nanostructures and make and design the specific properties we want them to achieve, and sometimes even properties which are not in nature. Some structures may even present nonlinear properties. Like in this picture, we see the split ring resonators, which we use in order to do some second harmonic generation, which is basically doubling the frequency of the light we shine it upon. So what we're doing, we use phase control of this metamaterial, and basically we use it to build a diffraction pattern, and by that we actually can split the light and shine it to the direction we would like it to go. So this enables us many things.
Next is number 19, Tamir Admon.
Hello, my name is Tamir Admon. I'm from Yael Roichman's group. Thanks for the opportunity to be here. My research is about information machine. The basic idea is that the machine converts information to energy. So now the basic principle, if we want to go from one state to another, we know from the second law that we need to apply at least direct work W on the system. And if you can see it, it equals at least to the difference in free energy between the two states. So what we wanted to do is if we have more information in the system, we can apply less work to get from one state to another. So a group from Japan added a phenomenological parameter, I, which represents the information in the system. And when it gets larger, we see that W can get smaller. And what we try to do here is build a system where there is a one-dimensional flow channel, and there is a particle that flows inside, and it also can diffuse. So what prevents it from going to the right side of the slide is a wall, and you can see in a black line the trajectory of the particle, and when it gets far enough from the wall, we move the wall towards the particle, and then on average, the particle's trajectory gets a little upstream rather than downstream. So what we did was simulations. Here you can see the diffusion of the particle, and then if we wait a long time, we can do log on the particles placement and get from Boltzmann factor the potential in the system. After that, we also tried moving the wall and get energy from it. You can see in the right graph, in green is the work that we applied, and it is 5 orders of magnitude smaller than the actual difference in free energy in red, and the yield is about 100%, but everything is perfect in simulation. And what we also did was start the experiment. We have a PDMS flow channel, and the particles position probability is in blue. The green is actual one D array of optical tweezers, and it acts as a soft wall, so it has a Gaussian potential. And you can see that it really can't cross the wall, and we just need to start the experiment. Thank you for your attention.
Next is number 20, Udi Binstock.
Okay, I'll use this. Hello, my name is Udi Binstock. I am at David Sprinzak Laboratory, and I will speak about how cells differentiate into patterns in a more of mathematical way. So, first of all, I will talk about biological, a bit of introduction about biological cells that begins with the lattice of disordered lattice of cells, which has no preference of being one kind of cell or another. And then a process is made, and in the final, we will get a very obvious pattern of two-kind cells. These two-kind cells could be lots of things. In this case, on the left, you can see a picture of the inner ear of a chick, and this inner ear differentiate into a pattern, a kind of a checkerboard pattern that includes air cells, which are light here, which are more bright, and between the hair cells, there are supporting cells, which are black in this case. So what is the process that made all of this? The process is called lateral inhibition, this by notch signaling. And what is the notch signaling? Okay, so the communication between the two cells, between either two cells, is being made by delivering proteins within the cell on the membrane of the cell, and then the proteins will connect to each other, and a process will make that I will just go over it right now, and then it will be differentiated. So as you can see here on the left, two proteins, they are called notch and delta. Notch is marked as green here and delta is red. They are going to be on the membrane of each cell producing both of them, and once they are connected, there is a complex, and this complex is being pulled by the cell that expressed notch protein, and this is the signal, this is the communication. This cut is this pull of the part of the notch that being cut goes back into the nucleus and tells the nucleus that this cell that expressed notch to produce less delta. All right, I hope you understand. You can come to my poster later. And then the cells that produce the higher value of delta will become air cells, and the cells in the end that will produce the lower value of delta protein will become supporting cells. Okay, so, sorry.
Okay, so the last one for this poster flash session is number 22, Yuval Tzur.
So today I'd like to present a poster in the field of electron beams and electron beam shaping. And thanks. My name is Yuval, and the work has been done under the supervision of Professor Adi Aryeh. We're going to show how to measure orbital angular momentum and the self-acceleration of electron beams. So in an electron transmission microscope, we look at the beam, which then illuminates a phase mask, which is inserted in the sample area, and we watch the diffraction pattern out of this phase or amplitude masks, and we record the amplitude of the Fourier transform, which is the diffraction pattern, we record it on a camera and observe the results. So one specifically interesting beam is the electron vortex beam. And this beam draws much attention due to its helical wavefront. And as you can see, the sample in B transfers the light into many different vortex beams, each of them carrying a different amount of angular momentum, which is, of course, quantized. And this is expected to open up in the future a new field in electron microscopy, which is called angular momentum spectroscopy. So this angular momentum is supposed to be exchanged with some sort of sample, maybe ferroelectric, but how can we measure the angular momentum afterwards? So that's a hint over there. We then take this method and we extend it also to another very interesting type of beam, which is the airy beam. It is being called non-diffractive and self-accelerating. This is, for example, the profile, the transverse profile of the airy beam, which we see in the electron microscope. And due to its chaotic shape, it accelerates without an external force in the outer plane, out from the paper. But then the question is, from that beam profile, how can we measure the acceleration with only one image? So the answer is probably also in the poster. And with that, I'd like to thank you very much. Enjoy the posters.
Okay, so, well, let's thank all the speakers of this session for their very nice presentations.